Okay, we're going to get started. So my name is Terry Moody, and this is the Translational Research in Clinical Oncology course. In the government, everything has an abbreviation, so this is abbreviated TRACO. And so here you see my vital statistics, and most importantly is the email. So that's the best way to reach me. And we have about 170 registrants, so our class is full. And on the organizing committee, I'd like to acknowledge Erwin Arias, Lupa Bardakovsky, and Farazia. Jonathan Wiest is the director of the Office of Training and Education in the NCI Center for Cancer Research. And so here is our schedule. TRACO routinely is Monday afternoons, 4 to 6. And we have it in class here in Building 50. We also video conference to NCI Frederick, Building 549, Room A. So we welcome all of our fellows at NCI Frederick. So basically the goal of TRACO is to discuss cancer research in translational terms. So for cancer, traditionally it's treated with radiation therapy. And so we have a lecture in radiation oncology in October. And also it's treated with chemotherapeutic drugs. And one such chemotherapeutic drug are the topoisomerase inhibitors. We have a lecture on that in late September. And we go through various types of cancer, such as the big killers in the U.S. are prostate cancer, lung cancer, breast cancer, and colon cancer. In terms of new therapies, one of the goals is to find tumors when they're very, very small. So we have a lecture focusing on tumor imaging. If you detect cancer when it's in stage 1, you can then have the surgeon go in and remove the tumor and alleviate the cancer entirely. As cancer progresses and grows bigger, then there's the process of angiogenesis, whereby tumor vessels from the host grow into the cancer. Subsequently, the cancer can undergo metastasis from the primary organ to distant organs. And in the US, a little over 500,000 people die from cancer, and almost all of them die from metastatic cancer. In terms of cancer, in some types of cancers, there's great genetic abnormalities. In addition, we worry about silencing of genes. So we have a lecture in epigenetics. And then in terms of new approaches in cancer, microRNA is now being studied, and some microRNAs function as tumor suppressors, some as factors that stimulate cancer growth. And nanotechnology, the primary goal of that is to deliver cancer drugs and minimize toxic side effects. So the course, there's no charge for this course, and the website is ccr.cancer.gov/careers/courses/TRACO. What we try to do is post the PowerPoints after the lecture, but this is somewhat difficult because the PowerPoints then have to be converted so that they are 508 compliant. Not an easy thing to do. And then one thing that differs from an academic course is we also have the possibility of visiting tumor boards where you see clinicians discuss patients, and core laboratory facilities. And this will be developed this month, and I'll be sending you all emails if you want to attend the tumor boards and or core facilities. And finally, when the class is done, you can get a certificate signed by our scientific director, and there's a computer-graded multiple choice exam that you have to pass. So are there any questions on the course organization at this point? If not, we'll move on to the lecture. And so we see then that in the US, the 4 big cancers are lung cancer, colon cancer, breast cancer, and prostate cancer. You see that each of these have an incidence of almost 200,000 patients per year. But in terms of treatment, breast cancer is readily treated. Only about 16% of the patients die. Similar numbers for prostate cancer. Colon cancer doesn't respond to treatment as well, and lung cancer responds to the traditional treatment very poorly, whereby over 90% of the patients that are diagnosed with the cancer will ultimately die for it. So we have a lot of work to do yet in lung cancer, and we see total about 40% of the patients diagnosed with cancer will ultimately die from it. So other types of cancer include pancreatic cancer. We have a lecture by Perez Hussein on this. And pancreatic cancer, it's also very difficult to treat like lung cancer, as is ovarian cancer and brain cancer. We have a lecture on lymphoma by Wyndham Wilson later on, and this cancer is readily treatable. And we also have a lecture on ovarian cancer by Tina Annunziata. So, what are some of the risk factors for cancer? Well, alcohol certainly is a risk factor for both esophageal cancer as well as liver cancer. Asbestos, which is insulation in the walls, that's a risk factor for lung cancer. Then the diet. This is a risk factor especially for stomach cancer. Some cancers are genetically based, such as colon cancer and breast cancer. Some cancers are affected by hormones, such as breast cancer is affected by estradiol. Prostate cancer is affected by androgens. And then obesity. This is a growing problem in the US. Over 30% of our population is obese now, and this contributes to colon cancer. Ionic radiation, such as the atomic bomb, contributed to leukemia in Japan. Tobacco is the big risk factor for lung cancer. Ultraviolet radiation is a big risk factor for melanoma. And cervical cancer is caused by viruses. So we see we have many different types of factors that can contribute to cancer. And my specialty is really lung cancer, so we'll discuss this initially. And in the US, there's about 45 million current smokers, but only 1 out of 6 of these people will actually die from lung cancer. Others die from things such as heart disease. When people stop smoking, that's good. 45 million ex-smokers are in the US. And when you stop smoking, there's an immediate benefit in terms of your risk to heart disease. However, it takes about 10 years to go back to normal risk for lung cancer. So when you stop smoking, initially you're still at very high risk to get lung cancer. And finally, it's difficult to quit smoking due to nicotine addiction. So, in cigarette smoke, there's many carcinogens, such as we'll be talking about polyaromatic hydrocarbons, such as Benz pyrene. Another factor is NNK. And then you can have other chemicals such as ethyl carbamate. This is paint varnish. And certain heavy metals such as chromium contribute to lung cancer. So in terms of the polyaromatic hydrocarbons, the benzopyrene is metabolized to the chemical BPDE, and then this in turn reacts with DNA, leading to mutations in the DNA. And especially in lung cancer, the genes for p53 and k-ras are mutated. So this shows the chemical of benzopyrene. It's an organic chemical. We have 3 benzene rings, 5 aromatic rings total. So it's very hydrophobic. And then the benzopyrene gets metabolized by enzymes such as P450 to benzopyrene 7,8 oxide, then a benzopyrene 7,8 diol by the epoxide hydrolase. And then in turn, this gets metabolized to BPDE, which is the carcinogen, and then it can form adducts with the DNA. So cytochrome P450 enzymes then catalyze adding oxygen to the benzopyrene, but then in turn phase 2 enzymes can metabolize the carcinogen to a water-soluble metabolite that can be excreted. So phase 2 enzymes are good, phase 1 enzymes are bad. So the DNA gets mutated then if the rate of carcinogen activation exceeds the rate of carcinogen detoxification and or DNA repair. And DNA adducts as well as inter and interstrand DNA crosslinks are removed by nucleotide excision repair. So P53, this is a tumor suppressor gene, and it mediates G1 to S phase transitions in the cell cycle. And it drives programmed cell death, or apoptosis, after the DNA is damaged. But if the P53 then gets mutated, the DNA damage is not repaired, and there's no programmed cell death, so the cancer cells grow. So DNA is increased along with the cell cycle inhibitor P21 after DNA damage, and phosphorylated DNA induces the expression of Bach's apoptosis. So P53 mutations are detected in most lung cancer patients, and consequently the G to T transversions occur at specific areas in the DNA, such as exon 5, exon 7, and exon 8. And there's an increase in G to T transversions in smokers relative to non-smokers. So here we see the point mutation percentage as a function of nucleotide, and we find that hotspots include codons 157 and 58, codons 245, 248, 249, and 273. So these mutations are not random in the DNA, they're at specific points. So in the cell cycle then, we have the G1 phase going to the S phase. This is regulated by P53. In the S phase, we have DNA replication. The G2 phase, additional enzymes are made so that when you get to the M phase, the chromosomes can be condensed and then segregated. So we have the mother cells leading to the production of the daughter cells. So cancer, it's growing continually. There's no rest in G0 phase. The cancer cells grow rapidly. And so P53 mediates the G1 to S phase checkpoint. And when there's DNA damage, there's an increase in P53 mutations as well as P21. And P53 drives programmed cell death after DNA damage. But if P53 is mutated, then the programmed cell death does not occur. So in terms of cell cycle enzymes, the big one is cyclin D as well as cyclin-dependent kinases. We see in the S phase, We have cyclin D1 going over to cyclin A1, and cyclin-dependent kinase 4 and 5 are turned on in the G phase. Cyclin-dependent kinase 2 is turned on in the S phase. In the G2 phase, then, cyclin A is still expressed, but cyclin-dependent kinase 1 becomes expressed, and not cyclin-dependent kinase 2. In the M phase, we go from cyclin A to cyclin B. Cyclin-dependent kinase 1 is still expressed. So in the cell cycle, then, the cyclins change as well as the cyclin-dependent kinases, and especially cyclin D. It's inhibited by P21, P27, P57, P15, 16, 18, and 19. So in terms of tobacco smoke exposure, it takes about 30 years for a patient smoking cigarettes to get lung cancer. And after 10 years, normal lung tissue undergoes hyperplasia and metaplasia. After 15 years, dysplasia can result. After 20 years, a carcinoma in situ can form. And after about 25 years, a malignant cancer can form. So we see we have normal epithelial cells in the lung. Then when there's hyperplasia, the epithelial cells start to grow. With dysplasia, the cells become disordered. With a carcinoma, then, some of the cells are changed genetically. And then with further genetic changes, the carcinomas become malignant and can invade other organs. So this is just a H&E slide illustrating staining of a lung section. This is the normal lung. You have epithelial cells, small dark nuclei, and we have an ordered structure here on the surface of the lung, and we see little villi on the surface of the epithelial cells. And these exchange the gases, and when you breathe in oxygen, you excrete CO2. So when there's hyperplasia, we still have some villi at the surface, but we now see that the epithelial cells are growing. With dysplasia, the cells now are becoming disorganized. We still see some cilia on the surface, but not as much as we used to see. And then when there's an adenoma, the nuclei become large. This is a benign tumor at this point. And then with further progression, adenocarcinoma forms. And this adenocarcinoma, you see very large nuclei, very little pink cilia, or pink cytoplasm. And these adenocarcinomas excrete various fluids, such as mucus. So again, here's a little cartoon illustrating that with carcinogen exposure, you get initiated cells, then with further promotion, these cells then form a small tumor, and then with further progression and mutation, a red carcinoma forms. So the carcinoma, initially it starts in the lung, but then when the tumor gets to be about 1 millimeter in size, it needs additional oxygen and nutrients. So the host blood vessels then infuse into the tumor, and the tumor becomes larger. And then when the tumor becomes very large, it can migrate from the primary organ, the lung, to additional organs. And it can invade and undergo metastasis. So for the lung, we have metastasis from the lung to lymph nodes, and the liver, and the bone, and then ultimately the brain. And genetic abnormalities in lung cancer include mutation of tumor suppressor genes such as P53, silencing of tumor suppressor genes such as P16 and retinoblastoma, and amplification of oncogenes such as c-myc, cyclin D1, EGF receptor, and ERB3. So it's thought in most epithelial cancers, like lung cancer, you have dozens of mutations of genes. And then the question is, what are the driver genes, the most important genes in the cancer development? And certainly P53 is one of these, and the EGF receptor is another one that's quite important. So we'll take a look at some of the tyrosine kinase receptors. And this has been the big advance in the past decade, is they've come up with many tyrosine kinase inhibitors, and the patients initially are very responsive to these drugs. So in lung cancer, it was found that the EGF receptor is mutated, and when it's mutated, it's sensitive to tyrosine kinase inhibitors. So in this red area, this is where enzymatic activity resides, and the EGF receptor can tyrosine phosphorylate various substrates such as PI3 kinase and other things, as well as phosphorylating itself. And we note the EGF receptor in green. There's domains 1 and 3. This is where EGF binds. And domains 2 and 4, these are cysteine-rich domains. It crosses the membrane once. Then we have a large tyrosine kinase domain, and then a C-terminal tail. And it's the C-terminal tail of the EGF receptor that can get phosphorylated. In contrast, all cancer cells also have the IGF-1 receptor, and it's similar to the EGF receptor in that there's an outside domain that binds the ligand, in this case insulin-like growth factor 1. But the EGF receptor is a monomer, whereas the IGF-1 receptor, it's an alpha-2, beta-2 heterodimer. So the two alpha chains bind the IGF-1, and it's the beta chains that have the tyrosine kinase activity. And other receptors that are especially important is the VEGF receptor. This is the vascular endothelial cell growth factor receptor. This is very important in the angiogenic process. So the cancer cells make VEGF, and it's the VEGF receptor on the endothelial cells that cause the angiogenic process. And we note the structure here. It's similar to the EGF receptor in that there's an extracellular part that binds ligand, and then there's a kinase domain in the intracellular portion. Now, the interesting thing about the tyrosine kinase receptors is their ligands. They're dramatically different. For the EGF receptor, it can bind epidermal growth factor, transforming growth factor alpha. This is what we see in a lot of lung cancer cells. It can bind amphoregulin, beta-cellulin, heparin-binding EGF, epiregulin. So there's many different ligands that can potentially activate the EGF receptor. And another name for it is HER1 or ERB1. Now, in breast cancer, the big thing is ERB2. HER2 are new. And for therapy, we have a tyrosine kinase inhibitor that works quite good on the EGF receptor. In contrast, for the HER2 new, there's no known ligand. But what they can block it with is the antibody. And what's interesting is the ERB2 can actually form a heterodimer with the EGF receptor. So the EGF receptor, when it binds ligand, it can dimerize with itself, increasing tyrosine phosphorylation, or it can form a heterodimer with the HER2 new, forming a active complex. So the EGF receptor, it's actually got 621 extracellular amino acids, and we mentioned domains 1 and 3 are important for binding EGF. Then there's a single 24 amino acid transmembrane domain, and the 541 amino acid intracellular domain has tyrosine kinase activity. And what happens is when the EGF receptor gets turned on, it can ultimately phosphorylate itself. And it phosphorylates itself at tyrosine 1068, tyrosine 1086, tyrosine 1148, or tyrosine 1174. And what we especially measure in the lab all the time is the tyrosine 1068 phosphorylation. So the EGF binds with high affinity to the EGF receptor, as does TGF-alpha. And Ira Paston here at NIH, he makes a lot of Pseudomonas exotoxin conjugates. And so this conjugate can then bind to the EGF receptor. And as a result of internalization, the PE38 can then enter the cancer cells and actually kill them. So this is what it looks like on a Western blot. When you add EGF, you get especially the EGF receptor tyrosine phosphorylated. This is the phospholipase C gamma, and this is the phosphatidylinositol 3 kinase getting phosphorylated. And if you have the tyrosine kinase inhibitors like erlotinib, this blocks the tyrosine phosphorylation process. So in lung cancer, what's really unique about it is the EGF receptor gets mutated. In other types of cancer, this doesn't happen. But especially this mutation L858R occurs in about 15% of the patients. And consequently, these patients are very sensitive to the tyrosine kinase inhibitors, gefitinib and erlotinib. So traditionally, lung cancer has been treated with combination chemotherapy, but the 5-year survival rate is only 15%. So it's not very effective. So these tyrosine kinase inhibitors, they were very promising because it was found in patients with mutated receptors, the patients improved dramatically. And here you see some of the mutations that occur, especially the L858R. And then this affects MAP kinase activity, and as we shall see, AKT activity. So the signal transduction pathways are quite complex. We mentioned that in lung cancer, especially TGF-alpha is secreted. It binds to the EGF receptor. The EGF receptor can form heterodimers with HER2/neu and then activate this RAS, RAF, MEK cascade. So the first of this cascade is the RAS, and RAS by itself actually gets mutated in about 20% of the lung cancer patients. And RAS gets activated when it binds and forms a complex with GTP, but when RAS gets mutated, then this RAS-GTP complex cannot be degraded. Normally, it's degraded by GTPase activity to RAS-GDP. So we know then that the RAS-GTP stays along too long when the RAS is mutated, the k-ras, especially at codon 12, but we don't have any real good drugs to stop this process from occurring. So one of the new initiatives at NCI is to have the lab in Frederick, the Frederick National Lab, using RAS as a molecular target to try to come up with new ways for therapy. And possibly one way is just using siRNA to mutated k-ras. The second thing in the cascade is RAF. So RAS-GTP can then form a complex with RAF and activating it. And in lung cancer, RAF isn't readily mutated. However, in melanoma patients, about 60% of them have this mutation, BRAF V600E. When they have this mutation, they're then sensitive to this tyrosine kinase inhibitor, PLX4032. So there's an over 80% response rate in patients with metastatic melanoma. And both RAS and BRAF, they're driver mutations in several types of cancer. So they're key players. So RAF then in turn can phosphorylate another enzyme, MEK. So this is an enzyme cascade. And MEK1 and MEK2, inhibitors are being used in patients by this drug trimetinib. And also another MEK1, MEK2 inhibitor that's being tested is selumetamide. So what you see then is these tyrosine kinase inhibitors. They're used at the level of the receptor tyrosine kinase as well as downstream enzymes. So MEK1 and MEK2 then will ultimately phosphorylate ERK, and ERK can go into the nucleus and induce transcription factors such as Phos or MYC, which stimulate the growth then of the cancer cells. So here's a little cartoon showing the tyrosine kinase receptor dimerizing, stimulating RAS, RAF, then MEK, then ERK, and ERK can then enter the nucleus and stimulate expression of nuclear oncogenes, leading to increased cancer cell proliferation. So this is a molecular model then showing the EGF receptor, and we have domain 1, 2, 3, and 4. And then the receptor dimerizes, and when it binds EGF, it binds to domains 1 and 3. And we see there's a conformation change, especially in transmembrane domain 4, and ultimately that is what turns on the tyrosine kinase activity. So this area is rapidly advancing. They're learning the molecular structure now of the tyrosine kinase receptors, and this is leading to the development then of additional tyrosine kinase inhibitors. They can use molecular models to then develop new drugs that will fit into the tyrosine kinase inhibitor binding site. So the RAS pathway basically stimulates growth. Another thing that cancer cells do is they increase survival, and this is done through the PI3 kinase pathway. So when the EGF receptor binds, it causes phosphorylation of the PI3 kinase, and this causes phosphatidylinositol turnover. And in turn, the phosphatidylinositol metabolite can activate AKT, which increases cellular survival. So the PI3 kinase then, we have its alpha-beta structure, a catalytic 100 kilodalton subunit metabolizes PIP2 to PIP3, and the PI3 kinase is mutated in breast cancer patients, about 25%, brain cancer 27%, colon cancer 30%, stomach cancer 25%. So when these residues are mutated in the PI3 kinase, it results in a gain of enzymatic activity and more PIP3, which stimulates cancer cell survival. So another thing involved in the PI3 kinase is the enzyme phosphatase intensin homolog, PTEN. So it metabolizes the PIP3 that the PI3 kinase makes and metabolizes it back to PIP2. But the PTEN is mutated in about 13% of the breast cancer patients, resulting in an inability to convert PIP3, which is active, back to PIP2, which is inactive. So AKT then, it prevents apoptosis of cancer cells, and AKT gets phosphorylated at serine 473, and when it's phosphorylated then, it phosphorylates bad and caspase 9, preventing apoptosis, and AKT is mutated in a small percentage of breast cancer in colon cancer patients. And then AKT in turn can activate another downstream process, the mTOR, and mTOR is an enzyme that's inhibited by rapamycin. So we see then we have a growth factor cascade, and we have a apoptosis cascade. And what basically happens now is we're entering a state of molecular medicine, whereby the first thing that you do is you analyze a patient's tumor. And you see what is mutated. And the mutation then dictates the therapy. So this is an NCI protocol for lung cancer patients. And if they find that the EGF receptor is mutated based on the initial analysis of the biopsy specimen, then these patients are treated with erlotinib. If they find that the EGF receptor is normal, but RAS, RAF, or MEK are mutated, they then treat with AZD6244, which is a MEK inhibitor. If the EGF receptor is normal, RAS, RAF, and MEK are normal, then they look for PTEN mutations, PI3 kinase, or AKT. If they find that these are mutated, then they use the PI3 kinase inhibitor, MK2206, on the patient. If EGF receptor, RAS, and PI3 kinase are normal, and ERB2 is mutated or amplified, this especially happens in breast cancer, the patient gets treated with lapatinib. And then if the platelet-derived growth factor receptor is mutated or KIT is mutated, it's treated with the tyrosine kinase inhibitor, sunitinib. So we note here that all of these enzyme inhibitors are used depending upon what mutation the patient has. And in this particular protocol, about 25% of the patients had EGF receptor mutations, about 20% had RAS mutations, about 10% had PI3 kinase mutations, and about 5% had ERB2 mutations. And then when you're all done, you analyze the tumor again in the patient and see if it's the same or if the tumor has changed. And what's often found in cancer is a moving target. So you can treat a patient with a drug such as erlotinib, but then a year later the patient becomes resistant to that drug. And the reason is the tumor is mutated and it's no longer responsive to the tyrosine kinase inhibitor. So cancer isn't a stable disease. It's constantly changing. You're constantly getting more and more mutations. So when they find that patients have erlotinib or gefitinib resistance, about half of the time the EGF receptor has undergone a secondary mutation to T790M, and this is no longer responsive to the initial tyrosine kinase inhibitors. So what the pharmaceutical companies are doing now is generating a second generation of tyrosine kinase inhibitors that are effective in the EGF receptor with the T790M secondary mutation. So the initial example of tyrosine kinase inhibitors was done in chronic myelogenous leukemia patients, and they were sensitive to the tyrosine kinase inhibitor Gleevec. So CML patients, they don't have a mutation, but they have a translocation, and segments of chromosome 9 and 22 are fused, resulting in the BCR-ABL gene. This results in tyrosine kinase activity that is constitutively active, and the BCR-ABL tyrosine kinase is inhibited by Gleevec. So here we see chromosome 22 and chromosome 9. And we have the fused translocation of the BCR-ABL gene leading to the BCR-ABL fusion protein. And we see this analyzing the chromosomes. We see that chromosome 9 is increased after the fusion in size, whereas chromosome 22 is decreased. And so here we see the normal chromosome 22 and the mutated chromosome 22 with the BCR-ABL. It's smaller. In case of chromosome 9, this is the normal, this is the fused, it's larger. So in phase 1 clinical trials, Brian Drucker became famous for this. It was found that Gleevec was effective orally at a dose of 300 mg or greater. Dose-limiting toxicities included nausea, vomiting, edema, and rash. So in phase 1, in phase 2 clinical trials, 53 of 54 chemotherapy-resistant CML patients responded. And after a year, most of these patients were still doing well. And after 5 years, 89% of the patients still had progression-free survival. And so here we see the Gleevec. It's a small molecule, a tyrosine kinase inhibitor. We have lectures on small molecules in the middle of October. And so the Gleevec is shown in purple, and when it binds to the BCR-ABL, the tyrosine kinase activity is reduced. Normally, the ATP binds to the BCR-ABL, resulting in tyrosine phosphorylation and subsequent tumor growth. So here, this is a ribbon model in purple of the BCR-ABL, and the Gleevec binding is shown in green. However, they found after 5 years, 17% of the Gleevec patients became resistant to the drug. And in particular, there was a point mutation, T315I, near the ATP binding site, which impaired Gleevec interactions. So subsequently, ponatinib, another drug, is now being used for the mutated BCR-ABL. So this is a big problem now in cancer research, drug resistance. And last year at the American Association of Cancer Research meeting, there were many, many discussions about this drug resistance. So the tyrosine kinase inhibitors are very promising, but they don't really cure anything at this point. But they give you a lot of hope, and they can prolong patients' life. So here we see a whole bunch of tyrosine kinase activators that have been used, such as for CML. We mentioned the mutated gene is BCR-ABL. You can use imatinib or dasatinib. For breast cancer, HER2 is mutated, and the antibody Herceptin is effective, as well as the tyrosine kinase inhibitor lapatinib. For melanoma, BRAF is mutated, and a tyrosine kinase inhibitor PLX4032 is used. In gastrointestinal stromal tissue, c-KIT is the main player, and this can respond to imatinib or sunitinib, tyrosine kinase inhibitors. And in lung cancer, the EGF receptor is mutated, and erlotinib and gefitinib are the tyrosine kinase inhibitors that are commonly used. So there's a lot of hope now with these tyrosine kinase inhibitors, and you'll be hearing about them throughout the course. And we sort of end the first lecture on practical steps to prevent cancer. So one of the things you should always do is check your house for radon, such as I used to live out in the country on a house that was on a hill. And we lived on the top floor, but the basement, when we went to sell the house, it failed the radon test. And they told me that if you lived in the basement, it was equivalent to smoking 2 packs of cigarettes a day. So what they have now in most houses are pumps in the basement, and it just pumps air out of the basement into the air, and subsequently any radon which is released from rocks that your house is built on will be pushed out into the atmosphere. You should also check your house for asbestos. We mentioned asbestos is in a lot of walls, and even some of the buildings here at NIH still have asbestos in the walls. So you always want to check out your workplace to see what the precautions are there. And then we always want to have a good water system. There can be lots of carcinogens in water that can cause cancer. And another thing that you have to be concerned about is breathing polluted air, such as I lived in Los Angeles when I was a graduate student for 5 years. And in Pasadena, every day around 2 o'clock, a brown haze would move through Pasadena on its way to Riverside as the wind blew the smog of the city from the west to the east. And it felt like it was choking you, and you breathe, and it hurt to breathe. And then an hour or two later, the brown haze disappeared, and your breathing went back to normal. And they say if you live in an area like that, for every year you live, you lose about a half a year of your life because of the air as being so bad. But there's other places that are a lot worse than Los Angeles, such as Mexico City or even Beijing, China now. You want to protect your skin, especially in southern parts of the US. The UV shield in the atmosphere is being broken down. There's less and less ozone, and as a result, more of the sun's harmful rays come through the atmosphere. And so you have to wear sunscreen, especially in the south. There, Arizona, Texas, and Florida. There are many, many incidences of melanoma of the skin. Don't breathe in smoke. Well, lung cancer was discovered in chimney sweeps, people who clean chimneys. And it's just the coal has lots of carcinogens, such as the benzopyrene, and this is what leads to lung cancer. You want to exercise daily. You don't want to be obese. Such as myself, my big thing is bicycling. So this last weekend, I rode in what's called the Civil War century. So there were 2,000 riders, and you ride through the Civil War battlefields, such as Antietam and Gettysburg. And you keep in very good shape by cycling. You want to avoid pesticides, especially in terms of prostate cancer. It's found to be high in farmers who use pesticides in their fields. You want to eat fruits and vegetables. These have antioxidants. So we mentioned for a carcinogen to become active, you need lots of oxygen. So the antioxidants will reduce the carcinogens that you get metabolized. You want to reduce red meat consumption, especially fats lead to increased cancer. You want to eat fish, certain lipids, such as omega-3 fatty acids, prevent cancer. You want to drink alcohol in moderation. You don't want to get liver cancer. You want to avoid unnecessary x-rays, as well as other things, such as CT scans. So if you're exposed to too much radiation, this can result in lots of reactive oxygen species, which initiates DNA damage. And finally, you want to reduce infections. Kurt Harris will give a lecture later on in the course, and about 18% of the cancers are thought to result from response to inflammation. So inflammation results in lots of growth factors being produced that can stimulate cancer growth. And then finally, we have a couple references for this lecture, especially the lecture from Hanahan and Weinberg. This paper right here came out in 2011, and this has thousands of citations. It's sort of one of the good recent papers. And the second paper here just describes the use of Gleevec in BCR-ABL signaling pathways. So that'll do it for me, and I'd be happy to answer any questions that you have. Thank you. Any questions? Okay, well, in general, the tyrosine kinase inhibitors, they're sort of the big thing going on now in cancer. And it gives patients a lot of hope, such as lung cancer, for 4 decades. We've been using radiation and chemotherapy, and the improvement in the treatment really hasn't progressed that much. So with these tyrosine kinase inhibitors now, they give us a lot of hope in the cancers that are difficult to treat, especially the lung cancer, pancreatic cancer, ovarian cancer, and brain cancer. Yes?
So, thank you for your talk. As you mentioned, once the patient was treated by this kind of tyrosine kinase inhibitor, they will develop another mutation. So, usually, how long is this?
From the tyrosine kinase inhibitors, it's remarkable. It usually takes about 1 year. And so for lung cancer, the EGF receptor gets secondary mutations in about half of the patients. But then also the resistance develops because there are k-ras mutations as well. Okay, we'll move now to the second speaker. What we try to do in TRACO is we have a basic scientist like myself lecture to give the research perspective, and then we have a clinician like Jill Smith lecture to give the clinical perspective. So she's a professor emeritus at Penn State University, and her title is Translational Research, Bench to Bedside, Clinical Trials. Jill.
Is it on? Can you hear me? Okay, thank you.
So
I'll just use this.
And you can use this pointer.
Okay, well, good afternoon. How many PhD candidates or PhD postdocs do we have here? Okay. Do we have any MDs in the audience? Good, I'm not alone. Okay, thank you. We are an endangered species, especially when it comes to translational research. So today I'm going to give you some of the nuts and bolts about how you take your ideas from the bench to the bedside. I do have a disclosure in that I'm a co-inventor on a patent that I'm going to talk a little bit about some of the development of this today, and I've received some stocks for licensing this patent. So the objectives of what I'll be speaking about today are that we're going to understand how an idea is taken from the research lab to patient care, what are the steps in conducting clinical trials, what are some of the obstacles to overcome with drug development, and I'm going to give you some examples from my translational projects, also the pitfalls and the prize. So, starting off, the reason why all of you are in this room is because you have some dreams. You have some inspirations. You want to do science. You want to find the cure for this or that. Well, I had dreams too, and I still have dreams. My dream was that I was going to win the Nobel Prize. I haven't given up yet. But we all have dreams. And one of the people I admire was Nelson Mandela, who also won the Nobel Prize. And he said that there is no easy walk to freedom anywhere. And many of us will have to pass through the valley of the shadow of death again and again before we reach the mountaintop of our desires. So any of you who have been working in the lab, you know that it's not always easy, and you don't always get the right result that you want the first time through. But I'm telling you these things to encourage you not to give up, and you don't always get to the top of the mountain on your first try. It takes many times for most scientists. And then another person that I admire, who was not a scientist, but it was Edmund Hillary. And if any of you know who he is, he's the first person to summit Mount Everest. But the first time that Edmund Hillary tried to summit Mount Everest, he didn't make it. He failed, and most of his team died in the effort doing it. And he was speaking before an audience, and he had a big picture of Mount Everest behind him. And he turned around and he looked at that picture and he said, "Mount Everest, you have defeated me, but I will return and I will defeat you because you can't get any bigger, but I can." And then lastly, we just had celebration of Martin Luther King Jr. Day and his wonderful speech about "I have a dream." And we all have dreams. Martin Luther King had a dream. And he said that to hold all these truths equal, to be self-evident that all men are created equal. So we're all here today because we have dreams, we have inspirations. We want to be scientists, we want to help people, we want to make discoveries. It's not always that easy. And when we're trying to do research, and there's many of us out there doing research and working in the labs and doing preclinical research. However, one of the problems is this bottleneck of drug development. And very few discoveries or ideas actually make it through this bottleneck. The majority, there's a lot going on up here, but it's really only the novel, brilliant, breakthrough ideas that are going to make it. And we all want that to be our idea. We want that to be our discovery. And then there's another bottleneck, if I can say that being here. This could represent all of the principal investigators and all the postdocs and people trying to apply for funding down at the NIH. And as there's many rushing to the finish line here, there's very few that actually make it. And funding opportunities are becoming more and more difficult. There's more difficult getting drugs approved through the FDA. So that's what I say, this Washington bottleneck, not just the traffic that you have around here, but there's other issues. A couple years ago, the NCATS was developed by Francis Collins. And the reason for that, it was to establish a national center for advancing translational sciences. And in this institute, they had several key objectives, including finding therapeutic target validation, doing preclinical toxicology, biomarkers that were useful for predicting outcome, rescuing and repurposing old drugs, helping with clinical trial design, and even post-marketing. So these are the areas that we talk about in translational science and drug development that are important. So what do you need to get started? Well, first of all, you need an idea. And not just any old idea, but it has to be a brilliant idea. And as many of you know, sometimes this is still loading. You're coming up with the idea and it's formulating and maybe this, maybe that, but that's okay. You have to have an idea, and we all have these ideas. And then you have to have a hypothesis. Once you have this idea, you have to have a hypothesis, and most good research is based upon hypothesis-driven research. So then what are the phases of clinical trials? How are you going to do all this? Well, first of all, most trials and drugs, as they're developed, go through a series of steps. They start off with experiments in the laboratory, just as many of you are doing right now. You work with cells in culture, in vitro, to test your hypothesis. If that works, then you may move on and test it in animals to see if the hypothesis stands up in animals. And then once it's deemed to be safe in animals, then it goes into clinical trials in human subjects. And it goes through phases. Phase 1 is to test the safety, toxicity, and what's the right dose. Phase 2 is looking for efficacy. And phase 3 is you compare it to standard treatment. And phase 4 is usually post-marketing. There are different types of clinical trials. There's treatment trials, there's prevention trials, there's early detection trials, screening trials, diagnostic trials, Quality of life supportive study trials and genetic trials are a big area of research today. And the type of trial, it can be phase 1, 2, or 3. It can be a randomized or a non-randomized trial. It can be blinded or it can be open. It can have a placebo control or not. And it can be a pilot study or not a pilot study. So what is a phase 1 trial? This is the first step after you've completed your work in the animals, and it's the first in human experiments or trials. Usually it's a small trial, maybe 15 to 30 people. And the purpose of a phase 1 trial is really to determine if it's safe and to find out what dose is safe. How should the treatment be given? Should it be given by an injection? Can it be absorbed orally? Is it going to be transdermal? And then how does the treatment affect the body? Is there any toxicity associated with it? So the phase 1 isn't really to prove if it works. It's just to prove that you can actually give your treatment to a human being and it's safe. A phase 2 trial is the efficacy trial. Is it effective? Usually there's less than 100 people, but there could be more. You have to have a primary endpoint. This can be a stumbling block if you choose the wrong primary endpoint. So you want to make sure you choose the right primary endpoint when you design your phase 2 trial. Most phase 2 trials are blinded or unbiased, and it determines how the drug or compound works. And is it more effective than a placebo? And it usually doesn't compare it to other treatments at this stage. So as you can see, both the physician here is blinded and the patient or subject receiving it is also blinded. They don't know if this treatment is the active drug or if it's a sugar pill or placebo. There's a phase 3 trial. A phase 3 trial is larger, anywhere from 100 to 1,000 people. And there's an equal chance that you're going to be assigned in this trial to 1 or 2 or even more groups. The purpose of the phase 3 trial is to determine how the new treatment compares to the current standard treatment, or maybe a larger trial on how it compares to placebo. And then there's the phase 4 trial. And in the phase 4 trial, it's hundreds to thousands of people. Often vaccinations are tested this way. And usually it takes place after the drug or treatment has been approved. And it's to provide additional information on the drug's risks, benefits, and its optimal use. So this is just a summary of the phase 1, 2, and 3 trials, showing the number of subjects needed as you increase going up, the length of time it takes to do each one of these trials, the purpose, phase 1 mostly for safety, phase 2 mostly for efficacy, and phase 3 compares to other treatments, standard treatment, as well as safety, dosing, and efficacy. The number of drugs that actually get to each one of these stages seems to decrease as they go on. A lot of drugs are stopped in the phase 1 trials because they're found not to be safe. And so that's where many of the drugs stop. If they make it through phase 1, they may not be shown to be efficacious compared to a placebo, and they don't get developed. So that's why fewer and fewer drugs make it through that bottleneck. So in a randomized clinical trial, there's an equal chance that you're going to be assigned, the patients will be assigned to 1 or 2 groups. One gets the most widely accepted treatment or standard treatment, and then the other one gets the new treatment being tested, which of course we hope will have reason to believe that it's better than the standard treatment. It's important that the groups are equally distributed. You don't want to have all 80-year-olds in one group and 20-year-olds in the other group. But the way you do your randomization is based upon what your primary goal is and what you're looking at. So you want to make sure the randomization criteria will give you balanced population. And it provides the best way to prove efficacy of a new agent or an intervention. So with randomization, you start off with a population of people, and then typically we have randomization criteria to decide who goes in this group and who goes in that group. And in a Crohn's study that I did with inflammatory bowel disease, we randomized people based upon the area of their intestine that was involved with the inflammatory bowel disease, and also the severity, because we didn't want all the people who had severe disease to be in the control group and all the people, with mild disease in the treatment group if we were evaluating as our primary outcome the severity or improvement in their disease. So that needed to be balanced so that there's an equal distribution. So what is a pilot study, and what's the purpose of doing a pilot study? So a pilot study is usually a small study. It's usually the first study in a particular area where you have no idea how many patients you need to include in the study to show that it is effective. And so a pilot study will iron out some of the difficulties so that you can move into a larger trial. It helps also to provide a tentative or an arbitrary response rate, even though most pilot trials are open, so that the statistician can help calculate a sample size for moving into the phase 2 trial to compare significance over control. And then treatment trials involve people, for example, who have cancer, testing new treatments, new combinations of drugs, new approaches to surgery or radiotherapy, and it determines the most effective treatment for people who have cancer, tests the safety and efficacy of new agents or these interventions in our cancer patients. The prevention trials, they evaluate the effectiveness and ways to reduce cancer risk and to prevent recurrence of cancer. Some of these may enroll healthy patients at high risk for developing cancer and access new means to detecting cancer at an earlier stage in healthy people. So there's early detection and screening or diagnosis studies, and these early detection trials test ways to detect earlier the cancer, such as pap smears, mammograms, different blood tests. They want to see if this will affect the outcome. So most of these are outcome-based research. There are diagnostic trials to develop better tools to classifying types and phases of cancer and managing patient care. And usually they include people who have signs or symptoms. There's some other types of trials, including quality of life studies and supportive trials that may be aimed at comfort of the family or at their patients, or at the patients and how their quality of life is. And then, of course, as we're moving into the age of genetics, there's genetics trials to determine how the person's genetic makeup can help influence the detection, diagnosis, prognosis, and the treatment. It'll broaden our understanding of the causes of cancer and develop targets against the certain treatments of a genetic type in the tumor. Once we do all these trials, there's very important issues, including how are the patient's rights protected when they participate in clinical trials. And there are ethical and legal codes that govern the medical practice that apply to clinical trials. First, we have to have an informed consent. There has to be a review board, and Dr. Moody mentioned that you could possibly attend a review board, which includes a scientific review or an institutional review board, an IRB. There are also data safety monitoring boards that monitor a study once it's underway, especially a blinded study. So if one arm of the treatment is better than the other, or if they find that everybody in the treatment arm is getting better whereas the placebo is not, they may stop the study early and let everybody be treated. And that's up to the Data Safety Monitoring Board. Whereas the principal investigator will not know who's on the treatment and who's on the placebo. So these are how patients' rights are protected. So how do you do it? What are the nuts and bolts of going from your bench research to a clinical trial? First of all, you need to find a target if you're going to possibly do drug development, which is some of the area of research that I do. And there are many different targets out there for cancer therapy. We heard about some of them today with the tyrosine kinase inhibitors. I happen to work with G protein coupled receptors. But there are a number of different targets that you can choose. One of the targets that I chose was an opioid receptor, and the group I was working with, we discovered a compound or peptide that actually interacts with opioid receptors called opioid growth factor. And we found the receptor in the lab through which this peptide worked, and it was a novel receptor, and it was linked to growth rather than other classic types of mechanisms that opioid receptors work through. And we hypothesized that the OGF could inhibit the growth of pancreatic cancer or other cancers through interacting with its opioid receptor. So in order to test our hypothesis, we did preclinical experiments. And we started in the lab, and we grew pancreatic cancer cells in culture, and we tested the effects of the OGF on their growth. We also had to prove that the cancer cells had receptors for the OGF, and if you knock out or block the receptor, you could eliminate the effect. We had to then study the cell signaling and processes for treatment. And then we also, after we did that, we had to show that we had the same effect in an animal model. So this is just doing a cell culture experiment where we treated some pancreatic cancer cells with the OGF, and we showed that we could significantly inhibit the growth, and we could reverse that effect by using an antagonist at a concentration of the antagonist that had no effect by itself. And then we did pharmacokinetic studies and binding studies to determine what the receptor capacity was and the binding affinity of the receptors to show that it is a receptor-mediated mechanism. And then after we did this, and it's time, and this took many years, but to go from there into a human trial, you can't do that without getting approval from the FDA. In order to get approval from the FDA, you have to apply for an IND number, or an investigational new drug number. There's a process involved with that, and there's paperwork involved with that. This is a 1571 and 1572 forms, if any of you are familiar with that. Also, what you need to do is you need to write a protocol. You have to have the whole protocol, what you plan to do, what is your research plan. You have to write a consent form that the patient is going to sign, and then these things need to be approved by the institutional review board or the scientific review committee. After you do all that, you have to find a sponsor to help pay for your research. Get some research funding, apply to get some grants, and then you have to have a responsible principal investigator. And if you can't be the principal investigator, you team up with somebody who can carry out the project with you as part of the team. This involves also having a research nurse and a study coordinator. So it's a team effort to get this project going. And then once the project is approved, it's very important that it has to be registered on the clinicaltrials.gov website. If the project is not approved and uploaded on the website, you cannot publish it. So that's very important, and it has to be approved and uploaded before you enroll your first patient. So this is the process that you have to go through before you can even treat the first patient. This is an example, and you can pull this up online, of the FDA 1571 form. And this form needs to be submitted to the FDA every single time you communicate with them or send them a letter or a document. And once you submit your initial proposal, you will get an IND number. And this happens to be the IND number that I was given for my proposal. And number will go on every form that you submit to the FDA. And then down at the bottom here, there's a serial number, and the serial number will be in sequence. Every time you submit a letter or a protocol or a change in your consent form to the FDA, they need to be sequential. So it's form, this one would be form 001, then it's form 002, and so they go in order. Every time you submit something, this form must accompany it. Also on the 1571 form, and I'm sure you can't read this here, but it tells you down at the bottom, and you check one of these boxes, what are you submitting in this this report for? And it'll tell you down at the bottom that you're submitting either a new protocol, you're submitting a change in the consent form, you're submitting something else, a new investigator. So you check the box of why you're submitting this form. But every time you communicate with the FDA, it must accompany this form. The other form that is submitted when you're starting a protocol is the 1572 form. And that has a little bit more information in detail about the investigator and the team that's going to be carrying out this project. The investigator needs to include a copy of their curriculum vitae that needs to be signed and dated. And that has to be updated each year when the annual report is sent into the FDA. You need to have the name of the site where the research will be done, such as Pennsylvania State University, the National Institutes of Health. You need to give the name of the lab and the lab that will also be doing the blood work or carrying out any of the tissue specimens, and the directors of those labs and their contact information. The head director of the Institutional Review Board and the names and contact information. All of the sub-investigators on the team plus their curriculum vitae, and if they're carrying out seeing the patients, sometimes copies of their medical license. And then this form also needs to accompany a 1571, and it goes with the series. So getting back to some of the research that I did. So with pancreatic cancer, first of all, you have to have an idea. Well, knowing about pancreatic cancer, what's the problem? I knew that pancreatic cancer had a dismal prognosis, that the survival was only about 3 to 6 months from the time of diagnosis, and that there was no effective therapy. And I had discovered with my colleagues this compound that would work with pancreatic cancer. So what was the hypothesis? Well, I hypothesized that OGF could inhibit growth of pancreatic cancer cells and in nude mice, so it should be also effective in humans. So where do you start? So I had to start with a phase 1 trial. With the phase 1 trial, I wrote grants to the NIH and got funding. I had to apply the FDA, and I got my investigator new drug number. I had to apply to the IRB, and I got an approval from them to carry this out. But then you get to the point where you're thinking, okay, I did this in mice, and you can see when I put human pancreatic tumors in nude mice, and I treated them with 5 milligrams per kilogram of OGF, their tumors shrunk away. And that's wonderful. But how much am I supposed to give a 70-kilogram man? You know, it's like trying to get out your calculator, and you don't want to start too high because you don't want to hurt anybody. You don't know what's the safe dose. You don't want to go too low and have it not be effective. So a lot of this is just using your knowledge. We usually start lower, and we take it a step down. I went about 200-fold lower, and then you work your way up. But it is difficult. And then you have to decide, did I use the right dose? So the aims of my phase 1 study, which was a first-in-human trial, was to study the safety and toxicity of the OGF in humans. I wanted to determine what the MTD was. That's the maximum tolerated dose. I wanted to study the biological kinetics and the metabolism of OGF in humans. I didn't know what would be the best route of administration. I knew it was a peptide, so I couldn't give it orally, or it would be digested by gastric acid and peptidases, so I had to decide, was I going to give it subcutaneously or intravenously? And I wanted to evaluate the safety of giving this chronically to patients, not just acutely. So the eligibility criteria that we used, we treated just adult patients. We wanted to make sure that they had histologically confirmed cancer. These had to be patients that were not eligible for surgery, which could be curative, because we had no idea if this was going to have any effect. And we wanted patients that had a performance score, what we call the Karnofsky score, that they would at least live 3 months so that I would know if there was any treatment and how they were doing. I didn't want them to die after their first dose, and then I would worry that it was a safety issue of something that I had done. So we had other exclusion criteria, such as serious medical conditions and certain vital signs, and some laboratory tests just to include the patients or exclude the patients. And then what we did is a classic dose escalation study called the classic 3x3 design. And in this study design, you enter 3 people at 1 dose, and you decide what your criteria are going to be for toxicity based upon the common disease criteria for toxicity, which is posted on the NCI website. And if they get, say, a grade 3 or 4, you make the decision what's going to be your criteria for cutoff, and you're writing the protocol. So if no one has a toxic effect, then you go up to the next dose. If somebody has a side effect, then you can enter 3 more at that same dose and try again. So you're testing 6 people. And then if only 1 out of 6, has problems, then you can go up. So there's a mechanism that you can do. So this is called the classic 3x3 design. So knowing that I had given 5 milligrams per kilogram in my mouse, I actually backed it down and started with 25 micrograms per kilogram in a human being. And the very first patient that I treated with this had abdominal pain and ended up in the hospital that weekend with some nausea and vomiting. Although this was a person who had advanced metastatic pancreatic cancer. And I had to make a decision, was it what I did to the person, or could it have been their treatment? And my study could have stopped right then and there. But we decided to go ahead with the other 2 patients in the trial, and everybody did fine. So you do have to have some understanding of the disease process and have, work with clinicians who understand oncology and the disease itself. So I went ahead and pushed on, and we did fine, and we went on to the next dose. We went up to 50 and 75. And then when we got up above 250, we had 2 grade 3 toxicities with patients developing low blood pressure, and so you back it down. So we ended up, our maximum tolerated dose turned out to be 250 micrograms per kilogram. But that's the process of how you go about doing this and determining what's the right dose. The next thing we wanted to determine was what's the right route of administration, and we did this by measuring the OGF in the blood by radioimmunoassays, and we did this after we gave it by an intravenous infusion and also after we gave it by subcutaneous injections. And we had some problems with the solubility of this, trying to get it very concentrated to give it subcutaneously, and some of our patients were injecting themselves 4 times a day to try to get adequate dose. And we ended up choosing to go with the intravenous route instead, for the patient comfort primarily. So after we completed the phase 1 trial and we found that the treatment was safe, we determined the right dose that was well tolerated, then we wanted to go to the next step, which is a phase 2 clinical trial. And again, I had to get funding to do this, and I went through the orphan drug program at the FDA to get money to sponsor the trial. I had to get a new IRB protocol, but I was still doing it on the same FDA IND number because that was the number assigned to me for this drug. And with this study and with cancer patients, you know, we did not want to necessarily compare to a placebo. And with most cancer studies, you don't. We did decide to use the OGF at our maximum tolerated dose. What we did was that we chose patients. The FDA would only allow us to use patients who had either failed or refused standard therapy. I wanted to treat patients who were just diagnosed, but we don't always get to do exactly what we like. And because this drug had not been shown to have efficacy yet, we hadn't done the phase 2 trial, the FDA said, you don't know if it's going to work, so you can't compare it to, you know, a placebo. You just need to go ahead and use people who've already failed standard treatment. Unfortunately, once you fail standard treatment or even investigational treatment for pancreatic cancer, knowing that the survival is only 3 to 6 months, you don't have a whole lot of time left. So it was not the best study design, but it's what I was permitted to do. So the way we did it in order to show that our drug was effective is when patients ended up failing treatment and they were eligible to go on to hospice, we offered them this treatment and said, would you like to try this treatment or go on to hospice? And so the patients who chose to go on to treatment enrolled in our study. The other patients who chose to go to hospice, we served as controls, and we put survival as our primary endpoint. So because we couldn't follow CAT scans and things like that in people who were going on hospice. So our patients had failed standard therapy. That was our controls, and we wanted to have some survival if we could, so we could make a reasonable comparison. And we got baseline in our patients who were on the treatment, and every 4 weeks, labs, physical exam, we performed quality of life surveys, and we did CAT scans to look at tumor assessment size every 8 weeks. And as I said, our primary outcome was survival. We did have secondary outcomes, including the clinical benefit, which is a quality of life measure. And we measured the tumor size and response rate with the RECIST criteria, which, if you're not familiar with that, it's called Response Evaluation Criteria for Solid Tumors. And it's a measurement to see if they're shrinking at all with your treatment. And this is just an example of one of our patients who was on the treatment at baseline. And if you're not familiar with it, this is a CT scan showing the liver here and the kidneys in the back. The pancreas has this X. This is the tumor in the pancreas with the X going across it. And this is here and at 8 weeks, and the tumor is shrinking at 16 weeks and at 24 weeks. So we had some CAT scan response results here. And then compared to our survival data, we showed that the OGF significantly improved survival compared to those that elected to go onto the hospice. And most survival curves are shown by Kaplan-Meier method here. So after we finished our phase 2 trial, we wanted to know, can we make it even better? And so we went on and we wanted to do combination therapy. And so again, once you learn something in the clinic, then you go back to the lab again. And this was very important. So we went back into the lab and we took some pancreatic cancer cells and we said, okay, well, we know that OGF inhibits growth and some chemotherapy can inhibit growth in cancer cells too, but what if we add the two together? And so we did, and we got even a better effect. And I know this is kind of gruesome, but these are tumors grown in mice, and you can see that with the combination therapy, that tumor is much smaller. Also, with the chemotherapy, this mouse is kind of shrunken His skin has this mottled and peely appearance to it. He was not healthy. So chemotherapy does have off-target effects and can have some deleterious effects on human beings too. So this mouse kind of represented the toxic effects of the gemcitabine. However, when we gave the gemcitabine with the OGF, the mouse was healthy. So that implied to us that maybe the OGF was giving a protective effect against some of the cytotoxic and the toxicity that we were seeing with the gemcitabine. So with this information, we again went and got approval, and I won't go through everything, but we did then take it into a clinical trial. And the bottom line is that when we used the combination therapy, that we had marked improved survival 4.3-fold over 5-FU alone and 3.4-fold over gemcitabine alone, and even 2.1-fold over the OGF by itself. So this showed that, you know, in a stepwise fashion, you know, monotherapy, then many of our drugs today with cancer therapy and HIV therapy, we're using combination therapy because we get a better effect and less toxicity often. So the other thing I would like to mention is intellectual property. Wherever you work for, they own your property is the bottom line. But it's very important because we're all scientists here, and we want to go out and present our work at national meetings, and we want to send in that abstract. Remember, before you submit anything to make a public disclosure, even abstracts, if there's some new idea or invention that you have, talk to someone in your intellectual property department and file an invention disclosure. It's really important that you do that before you go out and make it public, because once you make it public, you cannot file a patent, okay? So those are just some important tips. And you know, many of us say, well, I don't really care about the patent and getting rich on drug development, but a company is not going to be interested in your research if they don't have the security for licensing for the patent. So even if you don't care about what happens beyond you, it's never going to get into human beings and be developed because the companies are not interested unless they have patent rights. So then you find a company and you try to license the patent. And hopefully the patent will issue and you move forward from there. So you know, this OGF has gone through those stages, and a company licensed the patent that Penn State had for it in January, and they're moving on to drug development and taking it into trials to get it approved by the FDA. So, but you have to go through the proper steps to get there. So what are some of the obstacles with translational research today? And I'm telling you this coming from the trenches and having been a principal investigator and getting my own funding and everything and working in the bench and trying to treat patients. Of course, money is a problem. And there's lack of funds or misuse of funds or disparity of funds, but it's always difficult. You know, you have to have the money to do the research. But don't be discouraged, because there's still money available for good ideas, innovative science. You know, you need to explain when you're writing your proposals why your idea is the most important thing on the earth and why you're the person to carry out this research. Those are all very important. You have to say what the impact of your science could be if it were to get beyond your lab and taken to humans and to patient care. And we are all part of the HHS system here, so it's all about human medical research. One of the problems, speaking to the clinicians there, is clinicians don't always get protected time to do the research that's needed, but when we're doing patient-related research, you need to have clinicians. And so this is a difficult area, but there's a big effort today to try to protect clinician scientists so that this sort of research can go forward. When indirect costs are given to universities from funding agencies, unfortunately, they're not allowed to cover malpractice insurance. You cannot do patient-related research without malpractice insurance, and that money has to come from somewhere. So either if the university doesn't cover it or if they make us cover it, we have to earn that money, usually by doing clinical care, to help pay for the malpractice insurance. So if I could change things, that would be one thing I would change, is to help indirect costs, take care of covering the malpractice insurance to do clinical trials. There also seems to be this big chiasm between industry and NIH and academia. And truly, in order for us to move science forward and to get these ideas from the bench out to the patients, we all have to work together. We all have to work together. There's always been difficulty with patient accrual into research studies. Patients say, am I going to be a guinea pig? They're all concerned that they're going to be a guinea pig, but every patient that I've ever enrolled in a study has always said, I didn't realize that this is what research is all about. This is wonderful. I'm getting excellent care. So I think that there's really a lack of understanding in the community and patients about what really is involved with research. And it's not to test them as a guinea pig per se, but to try to advance science and to come up with new cures and treatments. And also, there's no more one-man bands. We need to have team science. I could not have carried out the things that I did without a group of people, without the PhDs, without a community of people that all work together to make this project come to pass, and some of the other projects that I've done. So you have to, the PhDs have to work with the MDs need to work with the PhDs, because there's some things that the MDs understand with patient care, and there's some things that the PhDs understand about the basic science, and they're both extremely important in drug development. So in order to move forward in this field and for translational science, first and utmost, you need to transform your mind. You need to think outside of the box. You've got to be novel. You have to be innovative. Think about your ideas and your dreams and what's important about them and what makes you stand out in the crowd so that you can be that one idea that makes it through the bottle top. And don't be afraid to take some risks. If I had backed off when that one patient got nauseated after the first dose, this drug would never have gone forward to help other people. Yes, I mean, we're not going to put the safety of people in jeopardy, but you have to understand what you're doing and be willing to understand it and take some risks going forward in your science. And the bottom line is does research have any clinical relevance to help people? And I get a little choked up when I see this, because these are some of my pancreatic cancer patients who gave me permission to show their pictures because they're long-term survivors. And, without some of the work that was done, we wouldn't have them here today. So now what do we do? Getting back to my dream and the Nobel Prize and looking beyond me and beyond you, some people never get to see their dream come to pass in their lifetime. And, that's sad. But Helen Keller said that only truly happy people are the people who found a cause bigger than themselves to live for. And Martin Luther King didn't live to see the day when Obama was sworn in as president, but his dream was fulfilled. And the entire chapter of Hebrews in the Bible, of Hebrews 11, is built on people of faith. They never saw their dream come to pass, but they lived for a hope of something bigger than themselves. So maybe I won't get the Nobel Prize, but maybe somebody else that I planted a seed in, that trained in my lab, or I inspired, will go on to do that and will keep some of my dreams alive and will help the science go forward. But I'm still not going to give up wanting to get the Nobel Prize, but I learned that in order to become a Nobel laureate, I need to increase my chocolate consumption and move to either Switzerland or Sweden. So the odds go up if you do that. So in that note, in closing, thank you for your attention. Don't give up on your research. Have wonderful ideas and pursue them. Work together as a team. And thank you for your attention. Are there questions? Yes? It's an endogenous peptide. So the question was, what is the OGF drug that we worked with? Some of the neuroscientists that I worked with at Penn State were studying opioid receptors, and there's endogenous peptide metenkephalin that is what we call OGF. And so it was through some of their background work where they've discovered that it had growth inhibitory properties and actually its mechanism of action is working on a nuclear receptor to down-regulate DNA synthesis. Other questions? Yes?
I have a question more specific about the phase 1. So why is choosing patients that are looking at cancer and not just healthy patients, just the dose?
Not just normal patients?
Yeah,
Oh.
just healthy patients.
The question was, in a phase 1 trial, why did we use cancer patients rather than healthy controls? In some of the treatments that we're using for drug development where you may have a toxic drug and it's for cancer therapy, there's a question of whether you should give that to a healthy control. And also, I think it's better to know in the population that you're planning to use the drug in, how do they tolerate it? Because somebody who has advanced pancreatic cancer may not tolerate it as well as a healthy control. So, other questions? But you're right, you often use healthy volunteers, but some of these drugs we don't. Yes, sir?
How exactly do you go about finding a company to implement this idea?
The question was, how did I find a company to implement this idea? I'm trying to think how we found. I've gone through several it took several companies until I finally hit on one that actually licensed the patent. So several companies would contact me and say that they saw that my patent was issuing, or they were interested in our research. And I went through different, I don't want to say negotiating, with at least 3 or 4 different companies until we landed upon one that went ahead with it. There's a lot of venture capitalists out there. There's a lot of small companies out there. And often you have to start with a smaller company rather than big pharma because they're willing to take risks. And when you don't know yet how it's going to work out, and often even when I started negotiating with some of them was before the patents even issue. So, you know, there's nothing wrong with looking at those companies. Sometimes the university, I mean, at Penn State, they have an intellectual property department and they help with drug development. And so they help get your innovative ideas out there, and they sometimes have symposiums where they do innovative, you know, and they bring in industry to see what's going on at the university to try to foster that sort of, you know, relationship. So that's another way that it's done.
Was that all after the phase 1
Yes, so that's the other question, is when does a company get involved? Is it before or after the phase 1 and 2 trials? In the, I want to say, the good old days, companies used to jump on board when you had something exciting in the lab. With money and everything getting tighter and tighter, companies are less willing to take risks unless they know that you can maybe give this to humans and it's going to be safe. That's not across the board. I mean, there are still some companies out there, but they're more willing to get involved if you've already taken it through those stages, because it's less work for them to do. Doing clinical trials is very expensive, and if you've already completed the phase 1 trial or the phase 2 trial, they have less work to do to get it approved through the FDA. So there was another question? The question was the timeline. I don't want to discourage anybody. As far as like the first idea, and I'd say 1996 was, you know, back when we were working in animals, and then from the time of that, so we're going 15 years? Yeah. But, you know, it's already through into humans and moving forward. You know, so it does take a while, but don't be discouraged. There is a light at the end of the tunnel sometimes, so. Yes, ma'am. The question is with the patenting of the invention, did I have to tell them what I was working on and vice versa? Usually when you start negotiating with a company, you sign what they call a CDA, a confidential disclosure agreement. And they'll tell you what their company is interested in, and you must keep it confidential. And if you share anything with them about what you're working on in the lab that has not yet been published, they are obligated by law to keep that confidential too. So you always, and before you talk to any companies, always ask them to sign a confidentiality agreement. You don't spill the beans and tell them everything you've been doing in the lab and you've got the latest and greatest until they've signed that disclosure. And usually wherever you're working will put that into place for you through the university or wherever. So, any other questions? Okay, well, thank you for your attention.